This video is going to get us through Lab 6, which covers the scientific method. That's actually Lab 1 in your lab manual, but we're doing it the 6th week. And at this point, we should have already covered the scientific method in lecture, and so we're just going to coming back and we're refreshing our memory about what the scientific method actually is. One of the reasons that it's very important that you understand this is actually pertinent right now, and so I wanted to kind of share this observation. I see a lot of people online talking about how they don't trust anybody's opinions anymore, either the CDC or the World Health Organization, because they keep changing their minds about what we should and should not do. What I really want to stress to you guys is that's the whole point of science. Coronavirus SARS-CoV-2, which is the virus that causes COVID-19, is a brand new virus. We had never seen it prior to November, December of 2019. Since it was brand new, we didn't know how it was going to interact with the body. We didn't know how it was transmitted. We didn't know how it could be treated. We had to do science to figure out all of that sort of stuff. And as we're doing the science and changing our ideas, the recommendations change based on new evidence. So at first there had been this very small study that showed that chloroquine or hydroxychloroquine might be able to help people get better faster when it came to COVID-19. And then after we actually got numerous studies underway, it showed it doesn't actually help. It doesn't make people get better faster. It doesn't reduce the length of time they're in the ICU. It doesn't reduce the death rate. And so at that point, we stopped recommending hydroxychloroquine and chloroquine, those malarial treatments. We did science and we found that remdesivir, which is an antiviral, can reduce the length of time that a person is sick, and it can help a person get faster. More recently, they have found another treatment, dexamethasone, which is a steroid drug. It helps to reduce the symptoms and get people better faster. We at first thought this disease just affected the respiratory system, but as it turns out, it has huge impacts on every organ system, and it does that by making blood clot. And we couldn't have figured all of that stuff out had it not been for science. So if you're one of those people who thinks, oh, people are changing their minds, I don't want to listen to them. Well, if you're listening to a scientist, the reason they changed their mind is because we got new evidence, and that new evidence helps us make better decisions in terms of how we're going to treat things or how we're going to prevent things. Now, if you're listening to just some Joe Blow on Instagram or TikTok who's trying to tell you that you should drink disinfectant because the president told you to, that's not science. So please listen to the scientists like Dr. Fauci. They're the ones that are out there reading the research, conducting the research, and they're finding new treatment options, new preventative measures. They're explaining how masks work at preventing the spread and how it doesn't reduce the amount of oxygen that you breathe in or the amount of CO2 that accumulates in your bloodstream or any of that sort of stuff. Just listen to the scientists. Yes, we change our minds, but we're not doing it just because. We're doing it because we have new evidence, and the evidence points us in better directions. So I just really want you guys to understand that. Next up. This is all that stuff we talked about in lecture already, but just to kind of reiterate, whenever we're doing science, we always have this hypothesis, this idea of how different things are going to be interacting with each other. It is a statement, and yeah, like I said up there, not a question. A lot of times people come out of high school having been taught that a hypothesis is a question that you're testing, but it's not. It's a possible answer to a question that you're testing. Hypotheses need to be testable, they need to be falsifiable, which means you can disprove them. Although we tend to not like to use the word prove, because again, new science might disprove us later on. So whenever we're talking about hypotheses, we tend to use the words supported or rejected. The experiment that we ran either supported our hypothesis by providing us with data that showed a relationship that we thought might exist, or we rejected the hypothesis because our data showed that those two things might not actually be related to each other. So we support or reject, we don't really prove a hypothesis. Next, types of data that can be collected during experiments varies. If you're doing experiments in social sciences, psychology, sociology, that sort of stuff, they tend to collect qualitative data. And that data can still be very useful, it's just not as precise as quantitative data that we're going to talk about here in just a second. So I have just been in the process of going to a conference that's talking about students' finances, and one of the researchers was talking about her dissertation where she was trying to find out whether or not a course on personal finance help students out or not. And so some of the questions that she asked were, what did you find useful about this personal finance course, and what did you hate about this class or not find useful? Well, the data that she got back from the students, it's not graphable, but it's still very useful. They liked doing budgets, and they liked being taught how to apply for car loans and things like that. They didn't really hate anything about the class. And so again, it's still very useful data. It's just qualitative data, not quantitative. Quantitative data is a lot more precise. In the biological sciences, we tend to like quantitative data because you can graph it, you can measure it. Somebody else can get the same results that you got. So one of the videos, or actually I think the only video that I linked to for you guys for this chapter, is a Mark Rober video. He conducted an experiment on his own about, do drivers go out of their way to hit turtles on a road? And so he got little plastic turtles and tarantulas and snakes. If you haven't watched that video yet, go watch the video because it will help this out. But what he did was he collected data on how many drivers would actually swerve out of their way to run over a leaf, a spider, a snake, and a turtle, and he disproved his hypothesis because he had actual quantitative data about X number of drivers would swerve out of the road to hit the turtle. One of the things we're going to graph at the end of this chapter is how does temperature influence the heart rate of fish, and there does come a point where the temperature starts to actually kill the fish, and so that's quantitative data. You have an exact measurement that was taken by a thermometer, so you know what the temperature was when we calculated the heart rate of those fish. Next up, this is going to be starting on page 3 of the lab manual. The experiment that we usually conduct during this chapter is about fermentation, so I wanted to tell you a little bit about it. Some of your cells can ferment just a little bit. Your muscle cells can. They would prefer not to, but they can in the absence of oxygen. Like if you're working out really hard, you actually can shut off blood flow to certain muscles because the muscle contractions squeeze the blood vessels and prevent the blood from getting to that particular muscle. And so when that happens, that means the blood's not delivering oxygen, but your muscles still need ATP. So when they can't carry out aerobic respiration, they can switch over to fermentation. It's not as productive a metabolic process. It doesn't make as much ATP as aerobic respiration, but it's something that can keep the muscles contracting and not cramping up on you if that blood flow does get occluded for some reason. So the question that we're usually going to test as we run this lab is, does temperature affect the rate of fermentation for yeast? Now, yeast is going to be our stand-in because we can't just hook your muscles up to tensiometers to figure out whether or not they can contract during this process. Yeast is capable of both aerobic respiration, just like your muscle cells, and fermentation, so we're using it as a stand-in, although they carry out alcohol fermentation while your muscle cells carry out lactic acid fermentation. So the question does have a hypothesis that goes along with it. Your hypothesis can either be really broad, like I just said temperature affects the rate of fermentation, or you can try to make it more specific if you prefer, like as temperature increases, so will fermentation rate. It's your choice how you write your hypothesis, but as a general rule, the broader it is, the more you have an idea of what you're trying to test, and you're not biasing your results with what you think is actually going to happen. So this is a fermentation tube, or a respirometer is another thing that it's called. What we ordinarily do as we conduct this lab is we mix some yeast together with some molasses. The molasses provides the sugar, the glucose, that's C6H12O6, that is the reactant at the beginning of fermentation. Then the yeast can ferment that sugar and in the process produce alcohol and, more importantly for us right now, carbon dioxide. Now, if you have gases being produced in a liquid, the gases will of course rise, and it rises in this tube, and then depending on how much CO2 gets produced, we can measure the amount. So sometimes there's little lines on the respirometer, sometimes you have to hold rulers up, but what we would do is we would mix together the molasses and the yeast in at least 3 separate respirometers. We would then tilt this so that this tube was completely filled up with the yeast and molasses mixture. Then we would put one of these tubes in a refrigerator, which is going to represent our 4 degrees Celsius. We would leave one of these tubes at room temperature, which is going to represent our 23 degrees Celsius or 22 degrees Celsius. They listed at different temperatures in 2 different places in the lab manual. And then we would put one of these tubes into a water bath that is a little bit warmer. It's set at 40 degrees Celsius, which is a little bit warmer than our body temperature. Then we would let it sit for a while to allow the yeast to ferment the sugar that was in the molasses, and then after a period of time we would collect all of those tubes and we would measure the amount of CO2 that got produced by fermentation at the different temperatures, and then we would try to see, does temperature actually impact how fast the yeast can ferment. So something that I want to talk about, because it relates to how you would graph the data, is independent variables versus dependent variables. Your lab manual doesn't actually start to talk about this until page 7, but I want to talk about it now because it matters. The independent variable is what you think is going to be impacting something. It's what you have actual control over. So in this experiment, we controlled what temperature we put those fermentation tubes at, the refrigerator, room temperature, or the water bath. And so that means temperature is going to be our independent variable. Now, if we come back and we look at our hypothesis, it's also the thing that we were testing in our question, and it was the thing that we thought was going to affect what we were measuring off over here. So that's our independent variable. When you do a graph of your variables, the independent variable is going to go on the x-axis, which is the bottom part of the graph. Now, the dependent variable is what you're measuring. So what we were trying to measure was fermentation rate. What we're going to use as a stand-in for fermentation rate is how much carbon dioxide got produced by the yeast as it fermented. So we would measure the gas bubble in the straight part of the tube right here to measure how fast that yeast fermented the molasses, or the sugar in the molasses. That would be graphed on the y-axis. So here's the data that we're going to play with. This is data that I got from a past class. And how do I make that stupid little thing go away so that I can get what I want? Hold on. Let's see if it goes away if I do this. Why are you here? Go away, bar. Ha! Yay. Okay. Sorry. Once again, I'm old. Y'all can mock me on your own. Okay, so we're going to practice graphing this at the bottom of page 4. So we've got the y-axis and the x-axis. Your axes always need to be labeled. So your y-axis is once again going to be your independent variable, the thing you had control of. So for us, it's going to be temperature. I cannot write with a mouse. I really should get my special little pen out, although that doesn't make it that much better. Our y-axis is the dependent variable, which was the amount of CO2 that got produced, which again, that was our stand-in for how fast our yeast could actually ferment. So we only had 3 temperatures that we worked with. We had 4 degrees Celsius, which was our refrigeration temperature. We had 23 degrees Celsius, which was our room temperature. I'm going to put 22. And then we had 40 degrees Celsius, which was our water bath that was warmer. Then what we would do is look at the data that we collected. So tube A, that was at the refrigeration temperature, usually has either no gas bubble or a very tiny gas bubble. And so we would put a little dot right around 0. Let's see, I'm also going to try to just change or put some little lines off over here on the side. This is why I need graph paper as I'm doing this. But I'll just do in multiples of 5. So 5 millimeters of CO2, 10 millimeters of CO2, 15, I didn't give myself enough lines, ... and we'll pretend that this is our 80 up here. So we had 0 millimeters of mercury produced at the refrigeration temperature. At room temperature, we produced 15. So we would come up, and at 22 degrees, we produced 15 millimeters of mercury. Now, since we have 3 discrete temperatures, the best way for us to represent this data would be with a bar graph, and so this is my attempt at making a bar. Next, tube C, water bath, we went all the way up to 80 millimeters of mercury. And so there's how you would graph that data. Look how pretty my graph is. All right. Sorry. Next, we would answer questions. Now, the reason why we like to graph data is it provides a visual representation that usually hits a little bit harder than, you know, writing out paragraph after paragraph of here's what happens. You can see in 2 seconds that temperature definitely has an impact on how fast yeast can ferment, and you can also see by the shape of those bars that as it gets hotter, we produce more CO2, which means fermentation rate is going to be increased. Now, next we have some questions to answer on page 5. write a hypothesis for this experiment. So I did provide you with one of those on a previous slide. This is a nice broad one, but I also mentioned you could make a more specific one if you wanted to. Like maybe you thought warmer temperature would increase fermentation rate. That's a hypothesis. Maybe you thought colder temperatures would increase fermentation rate. That's a hypothesis. Your hypothesis was just what you thought was going to happen. So you can have various answers for that, but that was mine on the previous slide. Which fermentation tube is the control? The control is the one that you're not changing anything up on, and so since room temperature doesn't have, hotter or colder, that's the control in this case. Number 3, like I said up here on the slide, it really is a trick question, and that's because there are 2 kinds of variables, dependent and independent. I did talk about what those were on this slide. So in our fermentation experiment, the independent variable was the temperature because we had control over that, and that's what we were testing. The dependent variable is what we actually measured, so that was the amount of CO2 that got produced. Number 4, which substance formed by the yeast is measured to determine the rate of fermentation? We measured carbon dioxide because that was easier to see. You could, in theory, also measure the amount of alcohol that got produced, but you'd have to be able to do some chemistry to do that stuff, so CO2 is what we did. Number 5, what is the role of sugar in this experiment? So the sugar was coming from the molasses. That's the fuel that the yeast can actually ferment. It breaks it down so that it can make ATP. Number 6, comparing the mixture levels inside the fermentation tubes, does the data support or reject your hypothesis? So if I come back to my hypothesis, temperature affects the rate of fermentation, and then I look at my graph, clearly temperature does affect the rate of fermentation. So what I could say is the data supports the hypothesis that temperature affects the rate of fermentation. Now, if you had thought colder temperatures would increase fermentation, and so that was your hypothesis, you would say the data rejects the hypothesis, because clearly based on our data, colder temperatures did not increase the amount of fermentation. So your answers may vary depending on what your actual hypothesis was going to be. All right. Next thing that I want to mention, and we're going to flip over to page 7. What they do here is they start talking about this little fish. It's called a zebrafish. And instead of just testing on one fish, they had 5 fish, and they tested on 5. Now, the reason that they did that is to allow for replication. The yeast fermentation experiment that we were talking about a little bit ago, that one didn't have any replication. Usually just each group puts one tube in each one of the different temperatures. That means there's places where errors might happen, and your errors, you might not pick them up because you only tested it one time. When you replicate your results, in this case with 5 fish instead of just 1, it takes out some of the variability that might exist between different fish, because if you know anything, you know not every person is the same. Like for some people, Tylenol doesn't help their headaches, but for some it does. If you just test it on a person who Tylenol doesn't work for, you might think Tylenol is not a valid treatment option for headaches. So we want to test it on a lot of people, not just one person. So replication allows us to get those outliers out of the way, and it makes our data more robust. So whenever you have replication, what you want to do is average together all of the treatments for that one particular variable. So they're testing here the effect of temperature on the heart rate of fish. They did 0 degrees Celsius, 5, 10, they did it in 5-degree implements. So within the 0 degrees Celsius temperature, you have fish A, fish B, fish C, fish D, fish E. Well, you need to average together their heart rates. So basically you just add up those 5 columns and then divide by 5 for 0 degrees Celsius. If you do that correctly, you will get 19.8 is the average heart rate at 0 degrees Celsius. At 5 degrees Celsius, it's 20.4. At 10, it's 30.6. At 15, it's 35.8. At 20, it's 40.6. At 25, it's 47.6. At 30, it's 57.4. And 35, I need to talk about some stuff. So 35 degrees is starting to get really warm when it comes to water, because notice this is degrees Celsius, not degrees Fahrenheit. Those plus signs indicate that those fish died, and if they died, they do not have a heart rate. That means we can't include them in our calculations. So instead of adding up and dividing by 5, since we only have 2 live fish at 35 degrees Celsius, we add those 2 up and we divide by 2. That gets us a number of 58.5. After we do that, what we have to think about is, in this experiment, what are our variables? Because we have to know that before we can graph it. So what is the thing that the researchers had control of in this experiment? It's the temperature. So the temperature is going to be the independent variable. It's going to go down along the x-axis. What is the dependent variable? It's what they're measuring. And so they measured heart rate. And so that's what's going to go along the y-axis. Once we know that, we can go ahead and start to graph the data. So they gave you some graph paper on the next page, page 8. You can use that to try to make your graph nice and neat. Mine will not be that nice and neat because, well, y'all know me by now, and I'm just not that good at drawing on my computer screen. But once again, temp is going to be off over here. Heart rate is going to be off over here. This time they did give us nice increments. And so, you know, this would be 0, this would be 5, 10, 15, 20, 25, 30, 35 is as high as they went on the temperature. And then our heart rate is going to go from 20 to about 60. So I'm going to do these in tens. So there's 10, 20, 30, 40, 50, and there's 60. All right, now since we have a nice continuum this time instead of discrete data, instead of doing bar graphs, we could actually do a line graph on this one. So at 0 degrees Celsius, the average temperature was 19.8. So I'm going to place a dot right there. 5 degrees Celsius, we were at 20.4. That's just a little bit above that line right there. 10 degrees Celsius, we jump up to 30.6, which would be right about there. 15 degrees Celsius is 35.8, which should be there-ish. 40.6 for the next one. This is why you need graph paper, so you can make sure you're keeping your lines all nice and neat. 47.6 for the next one. 57.4. And then 58 is what we have. Now, since we were going to do this as a line graph, after you put your dots in there, you just kind of connect your lines all up. Now, if you were majors, we would be talking about standard deviations, more important calculations that get run in standard scientific experiments, but you guys are non-majors, and so this kind of hits our point home. And so we've got a decent graph, so we can start answering the next questions that we have. What is the relationship between temperature and heart rate as zebrafish? This is what's known as a direct relationship. So as one goes up, the other goes up, and you end up with a line that does pretty much what our line does. It goes like that. Are zebrafish warm-blooded or cold-blooded? Well, they want you to take a leap there to try to figure it out, but anytime the outside temperature changes something about your physiology this much, you're going to be cold-blooded. There's also the fact that all fish are cold-blooded, so in theory you just knew that from high school. Why did most of the fish die at 35 degrees Celsius? It's too hot for them. They're cold-blooded. They can't handle those hotter temperatures. Now, everything has set temperatures that it can live at. Like, you can't live in Antarctica, but penguins can. So everything has their own set temperatures, and 35 degrees Celsius is at their higher extreme, so they can't get hotter than that. The rest of that stuff that starts on page 9 continues through page 10 and 11 and 13. Your lead instructor says you don't have to worry about that, and so this concludes the scientific method. Make sure you take the quiz.